In the last lecture module, we talked about how ganglion cells can be divided into two different classes based on their feature extraction properties of on and off center. Now, other feature extraction properties that they manifest, I don't want to go into those at this time. Instead, what I want to do is turn my attention to another type of classification. Ganglion cells also fall into three classes. Each of those classes contains, well, two of them anyway, contain on and off center cells. P cells, parvocellular, small, or midget cells, are small ganglion cells that have small receptive fields. They exhibit selective sensitivity to specific wavelengths and exhibit a sustained response to stimulation. P cells function to analyze form and color. They exhibit medium conduction velocity, approximately 2 milliseconds, when they leave the retina and join the optic tract. P cells comprise about 80% of retinal ganglion cells, so a huge proportion. They process color and include cells exhibiting green on, green off, red on, red off color opponency. Additionally, giant melopsin expressing ganglion cells show yellow on opponency and form part of the parvocellular pathway as well. So you might be surprised that 80% of retinal ganglion cells are part of the parvocellular pathway, but you shouldn't be. These cells carry primarily information about fine detail and color. So they're getting most of their information from cones, and most of that from the fovea, which, as we saw, is disproportionately represented in ganglion cells. And so that's why we get that 80% number in the parvocellular pathway. This huge number of cells devoted to this relatively small area, along with other cells devoted to periphery senses as well. Our second class here are magnocellular, large or paracell cells, M cells, and they comprise about 10% of retinal ganglion cells. Magnocellular cells, or M cells, exhibit a burst of firing when light hits their receptive fields, but show a decrease in firing as time elapses. M cells are larger and have larger receptive fields. They exhibit a transient response to light within their receptive fields. M cells are relatively fast conducting at approximately 1.6 milliseconds. M cells function to analyze gross features and motions of objects in their receptive fields. And M and P cells form separate parallel processing pathways that are largely dissociable in the primary visual pathway. Now there's one other kind of cell, bistratified cells. And bistratified cells are very small and were not really discovered till around 1994. Bistratified cells fall into large field and small field cells in accordance with the size of their receptive fields. Small bistratified cells exhibit blue on, yellow off light responses. They don't exhibit any spatial opponency, no on or off center stuff. The cells are sometimes called K cells because they project onto the lateral geniculate nucleus coniocellular layers. They relay information between what was thought of as cells that were intercellular layers as opposed to processing layers in the visual system. Large field bistratified cells exhibit blue on and yellow off light responses, no spatial opponency, and together these two groups comprise about 10% of retinal ganglion cells. And they don't just project onto that next stop in our primary visual pathway, the lateral geniculate nucleus. Some of them also project onto the superior colliculus, and they're part of the eye movement system that controls our saccading when we're looking at a visual scene. So you might feel kind of overwhelmed by all the detail, but believe it or not, I'm really trying to walk a fine line between overwhelming detail and gross oversimplification. So for example, the discussion of ganglion cells is focusing on spatial feature extraction and motion, mentioning color a bit as well. And only a few of the 20 ganglion cell types are identified and talked about in the lecture. And so I'll quote from Marcus Meister and Mark Tessier-Lavan. One can envision each separate population of ganglion cells as sending a distinct neural representation of the visual field to the brain. In this view, the optic nerve conveys about 20 neural representations of the world that differ in polarity, so on and off, spatial resolution, fine or coarse, temporal responsiveness, sustained or transient, spectral filtering, broadband or dominated by red, green, or blue, and spectral sensitivity, and selectivity for other image features such as motion. So I'm trying to tell a story that's reasonably detailed, but not overwhelming. And hopefully that's what I'm doing now. But I realized that what we thought of as this very simple, straightforward, intuitive processing system that we use all the time without any effort at all is really, upon more careful investigation, a highly complex, heterogeneous, multiple processing stream that yields something that we perceive as very simple and easy.